Hello, my name is Tania Mandiangu, and I'm a medical laboratory science major. My project is entitled The Physiological Effects of Extracellular Osmolality Changes in Bovine Red Blood Cells. For animal health and productivity, livestock must have access to enough water. If the livestock water requirement is not met, then it can lead to dehydration. Dehydration happens when the livestock or bovine lack enough water in order to function correctly. Changes in osmolality can be used to assess and monitor dehydration. What is osmolality? Well, it's a measure of how concentrated a solute is in the bovine's blood. Terms like hypertonic, isotonic, and hypotonic are used to explain how various solutions can impact cells and organisms. Hypertonic means that the water rushes out of the cell, causing it to shrink in size. Isotonic means that the concentration of solutes are the same inside and outside of the cell. And hypotonic means that the water flows into the cell, making it swell. So in the case of dehydration, the bovine's blood is in a hypertonic state. A way that we used to test the osmolality in the bovine's red blood cells was through an osmotic fragility test. The osmotic fragility test determines how fragile or delicate red blood cells are to shifts in their environment, especially variations in solution concentrations. We place the red blood cells in a water and PBS or phosphate buffered saline mixture with a variety of concentrations. If the red blood cells rupture in fewer hypotonic solutions than normal, this implies greater fragility. If they rupture in more hypotonic solutions, it indicates they are less fragile. Another way we tested osmolality was through a hyperosmotic shock. We placed the red blood cells in a ringer solution with 300 milliosmolar, which is the standard, and another ringer solution with 900 milliosmolars. We ran the samples on the flow cytometer and used a stain called an XN5, which binds to a phospholipid called phosphatidylserine, or PS, which appears on the outside of a red blood cell during programmed cell death or apoptosis. A cell's stress response can be triggered if it is exposed to a hypertonic environment as part of its external surrounding. This stress can cause alterations in the physiology of the cell, which may eventually contribute to the activation of apoptotic pathways. The hypertonic environment has the ability to upset the usual balance of ions and molecules within the cell, so disrupting essential cellular functions. So in all, we would expose our red blood cells to stressors related to osmolality, as research regarding that topic is pretty limited. This research is important for the welfare of livestock in settings that may contribute to dehydration. Thank you.